Students, welcome back to your own channel Vedantu NEET English. This is Basavaraj Sir, your biology master teacher. Quickly, let me know in the chat before we start anything today if my voice is audible to all of you and the presentation behind me is visible to each and every one of you. Quickly, let me know in the chat. And also, green us in the chat if you're excited for today's class, that is your plant growth and development. Now, this is the last chapter, right? This is the last chapter from your plant physiology. And if you have been following the last two classes, that is photosynthesis in higher plants as well as respiration in plants, this chapter is going to be very easy. And with that being said, your entire major chunk of botany from your class 11th ends today. Right? It ends today, students. It ends today. So let me know in the comment section if I'm audible and visible to all of you, because this is a, what do I say, very easy chapter. Yes, it's a very easy chapter, but it demands a lot of concentration and a lot of memory power. I'll be honest with you. Especially the last part of this chapter, that is plant growth regulators. The plant growth regulators requires a lot of dedication and consistency for you to remember every single hormone. And if you ask me, sir, how many marks, right? How many marks do you get from this chapter? Can anyone guess? Last year in NEET 2024, there were 4 questions. There were 4 questions from this chapter. And this is in 2024. From your 2025 alone, 2023, there were around 5 questions. 5 questions. So easily, students, this chapter is for around 16 to 20 marks. 16 to 20 marks, students. And let me tell you, the questions are going to be very easy. They are very easy questions. They are very easy questions. It all depends on your preparation now. See, obviously the questions are easy. When I say easy questions, I mean they are direct questions. They are direct question. Direct line of NCERT they'll pick up and I'll tell you. So let's go with all the lines of NCERT today and also learn the concepts of previously before we start the plant growth regulators. So let me know in the chat if you are excited for today's class, because it's going to be amazing. It's going to be amazing class. We'll have a lot of fun and I will teach you every single concept. So let's start with the first one. Let's start with the first concept for today. That is growth. Now, obviously you would have heard about growth. You would have heard about growth from your first chapter of your class 11th, Living World. In Living World chapter itself, we learn about growth. That is, there are 2 types of growth. There are 2 types of growth. One is called as your intrinsic growth. The other one is called as your extrinsic growth. Extrinsic growth. Now, what is the difference? The difference is very simple. Your intrinsic growth is found in your living organisms. Living organisms. Yes, it is found in your living organisms. Now, what about extrinsic growth, students? Extrinsic growth can be seen where? In the case of your non-living things. In the case of your non-living things. Non-living things. Example, let me tell you a quick example. See, intrinsic growth means the growth which is happening from the inside the body. Right, we are growing from the inside. There is cell division and there is increase in cell size from the inside the body. That is called as your intrinsic growth. But when we come to extrinsic growth, it is a growth from the outside. Example, listen to me. How many of you visited Kashmir? Tell me in the chat. How many of you have visited Kashmir or any mountain areas, in a cold mountain areas? When you visit such cold mountain areas in the month of your November, December, January, February, when there is heavy snowfall, can we see accumulation of snow? Can we see accumulation of snow on the mountains? Yes, we will see accumulation of snow. Now, this is technically your extrinsic growth. This is what we call as extrinsic growth. So I believe you should know the difference between intrinsic and extrinsic growth. Done. But if I ask you to define growth, how do you define growth? What is growth? So listen to me very carefully, students. If you want to define the growth, that is, it is an It is an irreversible. It is an irreversible permanent change. It is an irreversible permanent change. Irreversible permanent change. You cannot reverse it back. I'll give you a quick example. Anyone from the chat, let's see. Let's say Savita from the chat. Savita, for example, when she was in class 3rd, when Savita was in class 3rd, she was around 3 feet height. Okay, 3 feet height. Now when she's in class 12th, she's around, let's say, 5 feet height. From 3 feet, she grew to 5 feet. Now, can she go back to 3 feet, being 3 feet again? Can she go back to being small? No. Is that growth? Yes, it is growth. It is a permanent, irreversible change. You cannot turn it back. Like whenever you want, you cannot go back to being 3 feet or 2 feet. You cannot. That is called as your permanent, irreversible growth. Now, one more famous example is, this is height. This is height. Now, what about body weight? For example, last year, this time I was 60 kgs. Last year, this time I was 60 kgs. Right now, I am 65 kgs. I am 65 kg. 65 kg. Now, from 60 kgs, I became 65 kgs. Is that growth? Is that growth? You might be telling me, sir, yes, there is increase in body weight, there is increase in cells, there is increase in size. Yes, it is growth. But what if I decide to go on a diet? What if I go to gym and I decrease my weight again? Is there a change? Yes, there is a change. So if there is a change, can I call it this 60 to 65 kgs a growth? Obviously not. It is not growth. It is not growth now. Similarly, there are other 2 examples which is given you in NCERT. 2 more examples. The first example is very simple. The first example they have given you in NCERT is swelling of wood. Swelling of wood. You take a piece of wood and you keep it in water. You keep it in water. This wood will swell up. Now, is that growth? No. Why? Because you take the same piece of wood and you keep it in sunlight again, it will dry up again, and as soon as the water is lost, the wood will shrink. So is it a growth? The absorption of water by the wood, increase in size of the wood, is it a growth? No. It is also not a growth. It is also not a growth. Done. Now what about increase in size of your guard cells? Guard cells. When the guard cells take up the water and they swell. When the guard cells, they take up the water. When the guard cells, they take up the water and they swell, right? They swell. They completely swell up. That's called as turgidity. That's called as turgidity. They become turgid. Is that growth? They're increasing size. Is that growth then? No. Because as soon as the water is lost, they become flaccid again and the stomata closes. So that also is not growth. It is also not growth. So by now you might have an understanding, right? You might have an understanding, Srinath, you might have an understanding that, sir, what is growth? Intrinsic and extrinsic growth. What is a growth? It is a irreversible change. It is an irreversible permanent change. You cannot go back. Any doubt in this particular topic? Ask me right now, quickly, go. I believe there should be no doubt right now. There should be no doubt right now. Nice. Now, there's one more concept called as germination. Germination. All of you would have heard about germination from all the way from your class 6th, 7th, 8th, 9th, 10th. But what is germination? Germination is nothing but growth of a seed. What is germination, students? Germination is nothing but growth of a seed. A seed is growing. It is dividing into, hypocotyle and epicotyle, plumule and radicle. So that is called as your growth of a seed. So germination is nothing but growth of a seed. See, this is a diagram from your NCERT. We have a seed which is germinating, that is growth of a seed. We have hypocotyle, this is the cotyledon. Then we have your hypocotyle, we have your radicle here, we have your radicle. Then we have epicotyle, then we have plumule on top, and that is the growth of a seed, also called as germination. Also called as what? Germination. Now students, can I teach you some beautiful scientific terms? Can I teach you some terms with respect to plant and growth now? Some terminologies. Some terminologies such as your plant growth. Plant growth is said to be unlimited or indeterminate. It is called as what? Unlimited or indeterminate. Students, this right here is a NEET PYQ. Yes, it might sound like a simple word, indeterminate, but that right there is a NEET previous year question. So in this chapter, you might expect a lot of questions from your last part, that is your plant growth regulators, but you will also have some questions from your starting part of it. So hence you cannot ignore any of them. Okay. For example, it is telling plant growth is unlimited or indeterminate. It means that plant can keep on growing. Plants can keep on growing. Very simple. Plants can keep on growing. Now you might ask me, sir, how? Why is plants so special? Why can't I keep on growing? Why did my height stop after class 12th? Why? You might be so jealous of plants, right? Because plants have a special tissue. Your plants have a very special tissue called as your meristematic tissue. Meristematic tissue. Because of this meristematic tissues, they can keep on growing. Now, in plants, they have two types of meristem primarily. One is called as your primary meristem. Primary meristem. Primary meristem. Other one is called as your secondary meristem. Primary meristem as well as your secondary meristem. Now, what is primary meristem responsible for, students? Primary meristem is responsible for the initial growth of a plant. When the plant is in nursery, when the plant is in your LKG, UKG, the plant is initially growing. That initial young plants, there we have the primary meristem. There we have the primary meristem in the young plants. In the young plants. Now, what about the secondary meristem? Secondary meristem is present in your older plants, which is responsible for your secondary growth. Secondary growth. For example, if you look at this particular diagram of your NCERT, do you see something called as your RAM, shoot apical meristem right here, and also sham or SAM, that is your shoot apical meristem? They are nothing but your primary meristem. They are the primary meristem, responsible for the primary growth of the plant. Responsible for the primary growth of the plant. You also have your vascular cambium here. Vascular cambium. Yes, vascular cambium of your secondary meristem. It is part of your secondary meristem. Students, I would want you to write this in your book. Open your textbook, read the lines of NCERT, understand the lines of NCERT, and make short notes in your NCERT itself. Okay, in NCERT itself. Now, students, very carefully, your vascular cambium is responsible for the increase in girth of plant or girth of stem. Can you see? There is increasing girth of the stem. That is the main responsibility of your secondary meristem. Primary meristem is responsible for the elongation of root and elongation of primary meristem. But secondary meristem is responsible for your increase in girth of the plant. Increase in girth of the plant. Done. That is your first point. Undeterminate or unlimited. Unlimited growth. Okay. Unlimited growth is done. Now, what if I tell you plant growth open? What is the meaning of that? If I tell you, students, plant growth is open. What is the meaning of this? Plant growth is open. It's very simple. The meaning of plant growth is open means that new cells are added constantly. Every single day new cells are added. That is, new cells are added. Sir, added constantly. Constantly new cells are added. Constantly. Okay? Constantly. Now, the third point. Third point, if I say plant growth is localized. Plant growth is localized. Now, what is the meaning of this? The meaning is very simple. That is, plant growth is restricted to a few specific areas. For example, root and shoot. Root and shoot. So, there will be elongation of initial elongation of root, initial elongation of your shoot. So, plant growth is always localized to some areas. Root and shoot typical meristem. But what about in the case of animals? What about in the case of animals? In the case of animals, the growth is uniform. Growth is uniform here. You will never see a human with long hands and short legs. Will you ever see that? One long neck will be there. You see that? No. Plant and animals growth are always uniform. We properly grow. We properly grow. Okay. But in the case of plants, do you see long roots? Long roots? Yes. Do you see very long shoots? Yes, you see. Okay. So that is important to understand. Plant growth is localized. Plant growth is localized. The fourth point here is, when it comes to plants, new organs are always added. They're always added. Now, what is the meaning of this? What do you mean by, sir, they keep on adding organs? Can they keep on adding organs? Can we keep on adding kidneys? More and more kidney? No. We only have 2 kidneys. We only have 1 heart. But in the case of plants, they can keep on adding new organs. Now tell me, which is the photosynthetic organ in the case of plants? Which is the photosynthetic organ? You will tell me, sir, it is the leaves. It is the leaves. So can they keep on adding leaves? Can they keep on adding more leaves? Yes, they can keep on adding more and more leaves. Now, which is the reproductive organ in plants? The reproductive organ in the case of plants is nothing but your flower. So can they keep on adding more and more flowers? Yes, they can keep on adding more and more flowers. They can keep on adding more and more flowers. That's all. So plant organs can be always added. Plant growth is localized. In root and shoot, plant growth is open. That is, new cells are added constantly. And also, plant growth is unlimited or indeterminate. It is indeterminate. I hope till here there should be no doubt. This is very easy, students. Simple it is. Simple. So the next concept is plant growth measurable? Can we measure the growth of a plant? Obviously, answer is yes. Obviously, the answer would be yes. You will be like, yes, sir, plant growth is measured. But when we speak about growth, what are we actually talking about here? Are we talking about increase in surface area? Are we talking about increase in leaves, increase in flower? What are we talking about? When we get into the depth of it, when we get into the depth of growth, aren't we always talking about increase in cell size? Honestly, when we get to technicality of it, aren't we talking about increase in the cell size of a particular plant? Right? And when we talk about increase in size, we are always mentioning the living part of the cell. Obviously, the living part of the cell is called as what? Protoplasm. Protoplasm. Yes, protoplasm. So when I talk about growth, of a plant, technically I am talking about growth of protoplasm. Yes, I am talking about growth of protoplasm. So can I call it protoplasmic growth? Protoplasmic growth? Yes, we are talking about action of protoplasm. That is the living part of the cell. Which is the living part of the cell? Living part of the cell. Living part of the cell. Now, but is it easy to measure protoplasmic growth? No. But can we measure the increase in protoplasm? Yes, we can measure. Yes, we can measure, but it is very, very difficult to measure. It is very difficult to measure. And that is the main reason. That is the main reason we have certain alternatives. We have certain alternatives. For example, the most famous alternative is increase in cell size and cell number. Increase in cell size and increase in cell number. When we talk about increase in cell number, I am talking about your root apical meristem. This is an example. This is an example. Root apical meristem of your maize. Root apical meristem of your maize. And if you want to calculate the increase in cell number, do you know how much is it? It is almost 17,500. 17,500 increase in cells per hour. Per hour. There is an increase in 17,500 of cells per hour. Can we count it? Yes, we can count it. And that's how we know the value. Is there growth? Yes, there is growth. Yes, there is growth here. Now, what about increase in cell size, students? The example is the cell of a watermelon. Watermelon. It is increasing in size. Yes, increasing in size. By how many times? Around 3.7 lakh times. 3.7 lakh times it is increasing the cell size in watermelon. So can we measure it? Yes, we can measure it. Yes, we can measure it. Now what about increase in fresh weight and dry weight? Obviously, fresh weight will be higher because of water content. Because of water content. Yes. Dry weight will be obviously comparatively lesser. Dry weight will obviously be a little bit lesser. Done. Now what about increase in length? Can we measure the increase in length of a particular structure? Yes. That is pollen tube growth. Pollen tube growth. Increase in pollen tube length, we can measure it. Yes, we can measure it. You can easily do this experiment at home. Take a pollen grains, add it onto a sucrose solution. If you add the pollen grains on a sucrose solution and observe it under the microscope, you can see the development of the pollen tube. Easy. Is there increase in surface area? Can we measure the increase in surface area? Yes. This is the leaf. This is the juvenile leaf. Now this leaf after some days will end up becoming very big. Can we measure the surface area? Yes. We can measure the surface area. We can easily measure the surface area of the leaf. Increase in surface area. Done. Increase in volume. Yes. We can measure the increase in volume. So what are we trying to establish here? We are trying to establish here that yes, growth can be measured. Growth can be measured. The actual measurement is what? Increase in the protoplasm or increase in the protoplasm. But can we easily do it? No, we cannot. Are there any alternatives? Yes, these are the alternatives. These are the alternatives which will help us to measure it. Help us to measure it. Clear, everyone? Tell me in the chat if this concept is clear to all of you. If this concept is clear to all of you, tell me in the chat quickly. Tell me in the chat quickly if this concept is clear. Because the next concept is going to be phases of growth. Phases of growth. Yes. Now students, all of you are growing. Yes. Once upon a time you were in nursery. Then you went to LKG, UKG, primary, secondary, college, degree. Are there different steps here? Yes. These different steps, can I call it different phases of your growth? Yes. These are the different phases of your growth. Similarly, when it comes to plants, there are different phases. Means there are different levels. And if you want to broadly classify the growth, it can be divided into three categories. The first category is called as meristematic growth. It's called as meristematic growth. Meristematic growth. Second, meristematic phase. Meristematic phase. The second one is called as your elongation phase. Elongation phase. And the third one is called as what? Maturation phase. It's called as maturation phase. These are the three different phases in a plant life. For example, Every single phase has certain properties. Every single phase will have a specific property which you need to know. For example, in your meristematic phase, first phase, it happens in your meristematic region. The cells are very small size here because it is the initial phase. The cells are very small here. Dense cytoplasm or protoplasm because they are the dividing cells. Initial stage. There's a dense protoplasm. Vacuoles are absent. Why do they need vacuole? Because first of all, there is no requirement of vacuoles in the initial stage. In initial stage of a growth, there are no products which they want to store. Vacuoles is the storage unit. If they don't have any metabolites, if they don't have any products, why do they need even storage? They don't need it. Conspicuous nucleus, yes. Cell wall is very primary because this is the initial stage. This is the initial stage, so the cell wall is very primary. The cell wall is a very primary cell wall. Now later what happens? Later we have the growth of the secondary cell wall. Then we have the secondary cell wall. Okay. So first primary cell wall, then we have the secondary cell wall. In the starting stage, in the first phase, we have the primary cell wall. And the last point, the very important point here is that multiple plasmodesmatal connections. What is this plasmodesmatal connections? Students, remember, let me teach you what is plasmodesmatal connection. If this is your cell, this is one cell, this is one more cell, and this is the cell wall. Obviously, the cell wall is very rigid. Yes, you'll be like, sir, the cell wall is very rigid. So how can two different cells interact? How can there be exchange of material? There can be, because of certain cytoplasmic connection. This is the cytoplasm. This is the cytoplasm of the cell. Because there are a lot of cytoplasmic connections, thin, very minute, very minute cytoplasmic connections. We have the cytoplasmic connections. Cytoplasmic connection. Right? Cytoplasmic connections. Now these cytoplasmic connections are called as what? They're called as your plasmodesmatal connections. And these plasmodesmatal connections, right, they will be present in the very young stage of the cell division. Okay, this is, since it's the first phase, a meristematic phase, here we have the multiple plasmodesmatal connections, also called as what? Cytoplasmic connections. They're called as cytoplasmic connections. Okay. Now students, they can directly ask you any of the following properties in the form of question. For example, when it comes to elongation phase, it's the second phase. In elongation phase, there is increase in cell length. Yes, cell length and increase in size because the cell is elongating now. And also vacuoles are present now. Vacuoles will appear because there is some amount of metabolite which needs to be stored now. So vacuoles we need vacuoles. Secondary wall formation is happening and depositions in the wall starts. That is, lignin depositions are starting. They are just starting. They are just starting here. Lignin deposition starts formation now. Okay. Now, the next third step is very important. The third stage is very important. That is, the third stage is called as your maturation phase. Now, once you have, I believe you might have had an option, right? After class 10th, you can take commerce, arts or science or any other field. You can take multiple fields. All of you took science. Means you decided your role is fixed now. You can go into any kind of scientific field. You can go. Unless you'll be like, no, I want to become a singer later on, you can become a singer also. But right now, in your mind, you have that I should become a doctor. Your goal is fixed. Similarly, in the maturation phase, the cell's function is fixed here. That is, in the maturation phase, there are two main things are happening. That is, first thing is happening in the cell wall. That is, in the cell wall there is lignin deposition, proper lignin deposits. Okay, And also there is change in protoplasm. Change in protoplasm. What is the change in protoplasm, students? In your protoplasm, every single cell right now has a fixed function. Okay. In your protoplasm, all the vacuoles, right? All the vacuoles are reaching maximum size. They're reaching the maximum size. All the vacuoles. There are vacuoles now in the maturation region. And every single cell here, every cell has fixed position. Fixed position. They have fixed position and they have a fixed work. Everyone's work is set now. Everyone's work is set. Okay. So any doubt here, ask me quickly. Any doubt here? Ask quickly. Where is the fixed position? Fixed structure and function. Fixed structure and function. Okay. Done. If that is done, can I teach you one more concept? If that is done, can I teach you one more concept called as something called as distal and proxial? They might ask you a question here. Distal and proxial. For example, this is the soil. This is the soil. This is the taproot. This is a taproot. Simple taproot. This taproot has three regions. This is called as region of maturation. Maturation. This is region of elongation. Region of elongation. This is region of maturation. Region of maturation. Region of maturation. Okay. Now keeping soil as the reference, I can call this region as distal region. Okay. And this is the proximal region. Okay, this is the proximal region. Okay, this is the proximal region. Now if I ask you a question, students, which is the proximal region to region of elongation? You should be able to tell me, sir, proximal region to elongation is maturation. Which is the distal region here? Distal region is going to be maturation. Now if I ask you a question, which is the distal region to maturation? The distal region is going to be elongation. For elongation, for your maturation, for a region of maturation, which is the proximal region is going to be elongation. So based on the reference to the soil, we can tell proximal region and distal region. Proximal and distal region. Okay. Proximal and distal region. Now this is a diagram of your NCERT. This is the diagram of your NCERT. Can you see? This region is your maturation region. Region of maturation. Region of elongation. Region of elongation. This is region of maturation. Keeping your root tip as an example, you can divide the entire different regions of a root tip. Okay. See this question, all of you. See this question, all of you. The question is very simple from your NEET 2023 question. Which of the following statements is not correct? Okay. The phase of cell elongation of plant cell is characterized by the increase in vacuolation. In elongation region, is there increase in vacuoles? Did we learn that? Vacuoles are present? Yes, vacuoles are increasing. Okay, correct statement. Now what do we have? The cells in meristematic phase growth exhibit abundance of plasmodesmal connection. Yes. Plant growth is generally determinant. Plant growth is measurable. Yes, we can measure the plant growth. But do you think the plant growth is determinate or indeterminate? The plant growth is indeterminate, right? It is unlimited growth. So which is the wrong statement here? The wrong statement is going to be option number C. That is your wrong statement. Wrong statement. Okay, that is going to be your wrong statement. So you should be able to tell such questions with very ease. So let's see if you have covered every line of NCERT now. See here. Done. We learnt about your did we learn about growth here? Intrinsic growth, internal, extrinsic growth, external growth, right? External growth. External growth is seen? Yes, it is seen, sir. Then, did we learn about your germination? What is germination? Germination is nothing but growth of a seed. Yes, growth of a seed, right? Growth of a seed. See, what is growth? Growth is an defined as irreversible permanent increase in size. Yes, it is an irreversible change. It is an irreversible change here. Okay. Now, one more thing is you need to know that it is both catabolic and anabolic in nature. Catabolic and anabolic in nature. Okay. See here, expansion of leaf is growth. How do you think swelling of piece of wood? Tell me, is swelling of piece of wood, is it growth? No, it is not growth, because a wood which is swollen can come back to normal as soon as we keep it in sunlight. As soon as we keep it in sunlight, right? One second, students. As soon as we keep it in sunlight. Okay. So, plant growth is indeterminate. What is the meaning of indeterminate? That is unlimited growth. Plant growth is completely unlimited. So, unlimited growth throughout their life. This ability is because of the presence of meristematic tissue. Because of meristematic tissue. We have primary meristem and secondary meristem. Primary meristem and secondary meristem. Okay. This form of growth where the new cells are always added. Did we learn that? New cells are always being added to the plant body. Is the activity of meristem is called as open. Did we learn that? In open growth, see, this is a line of NCERT, students. This right here is a line of NCERT which many students don't read. Do you see here? It's called as open growth. It's called as open growth. That is, new cells are always added. Okay. Now, we learnt about your root apical meristem and as well as shoot apical meristem. Shoot and root apical meristem. Done. This is your root apical meristem, shoot apical meristem, vascular cambium, which is your secondary growth. Secondary growth. Right. Now, in dicot plants and gymnosperms, the lateral meristem, vascular cambium and core cambium appear later in life. They are your secondary meristem. Secondary meristem. They cause the increase in girth of the organs in which they are active. That is, increase in girth of the plant stem mostly here. This is called as your secondary growth of the plant. Secondary growth of the plant. Now, is growth measurable? Yes, the growth is measurable. Now, how do you measure growth, students? Growth is actually measured by the increase in the protoplasm. Right? Actual growth. See here it says growth at cellular level is principally consequence of increase in the amount of protoplasm. Did we learn that? Yes, we learned that. Increase in the protoplasm. But can we measure the protoplasmic growth? No. It is very difficult to measure. We can measure it, but it is very difficult to measure. Very difficult to measure. So we have, since increase in protoplasm is difficult to measure directly, one generally has some other alternatives. What is that? Growth is therefore measured by the variety of parameters such as increase in fresh weight and dry weight. We learned that. Increase in length, area and volume. Yes, we learned that. And increase in cell number. You may have find it amazing to know that a single maize root apical meristem, RAM, can give rise to more than 17,500 new cells per hour. We learned that 17,000 cells per hour can be given from a root apical meristem of a maize. Okay. Similarly, if you can see here, in the form of the growth is expressed here, where is it? Whereas cells in the watermelon may increase size up to 350,000 times. 350,000 times. Done. Now, what about growth of the pollen tube is measured in terms of its length. That is measured in terms of length. That is pollen tube growth. Now we also learned increase in the surface area denotes the growth in the dorsal leaf. We learned, right? Increase in the surface area of the leaf. Surface area of the leaf. That growth can be measured. Growth can be measured. Then we have phase of growth. Phases of growth. There are three different phases, right? There are three different phases. First one is called as what? Region of meristematic. See, namely meristematic region, elongation and maturation. They are the three different phases. Three different phases of growth. Three different phases of growth. We learned that. Looking at root tip, this is your meristematic, elongation and maturation, as well as your maturation. Done. Okay. Now, here the properties are. The cells in the region which are rich in protoplasm possess large conspicuous nuclei. The cell wall are primary in nature. We wrote this. Meristematic regions, the cells are primary in nature and thin and cellulose with abundant plasmodesmatal connections. Every single point I have given you in the form of points, so it will be easy for you to remember and easy for you to read. And every single line of NCERT has been covered till now, so you do not miss out any single point from this chapter. You cannot miss. Okay. See, the cells proximal, just next away from the tip to the meristematic region, represent the phase of elongation. Elongation. Now, what happens in elongation? Increase in vacuolization, that is, increase in vacuoles. Increase in vacuoles, right? Increase in vacuoles. Then what is happening? Cell enlargement, a new cell wall depositions are also characteristics of your cells in this phase. Furthermore, from the apex, more proximal if you go, the phase of elongation we have. In elongation, what happens? Lies the portion of axis which is undergoing phase of maturation. In maturation, we have what? In protoplasmic modification, we have the vacuoles become maximal size. And here, most of the tissues and cell type you have studied in earlier classes present in this phase. Everything is from this phase. That is, in region of meristem. In the region of meristem, every cell has fixed function and structure. They have fixed function and structure. Done. Fixed function and structure. Where? In the region of maturation. In the region of maturation. Clear? Students, every single line has been covered till now. Every single line. Now, next concept is going to be growth rate now. Next concept is going to be growth rate. Okay? So, before we go to growth rate, let's finish growth rate in our terms and then we will go to growth rate there. Then we will confirm with NCERT. Okay? What is growth rate? Growth rate is nothing but growth per unit time. Growth per unit time is called as your growth rate. Growth per unit time is called as your growth rate. Okay. Now, if you look at your NCERT, there are two types of growth. The first type of growth is called as arithmetic growth. It is called as what? Arithmetic growth. Arithmetic growth. The other one is called as your geometric growth. Geometric growth. Arithmetic and geometric growth. Now, what happens in arithmetic growth, students? Listen to me very carefully. Listen to me very carefully now, students. In arithmetic growth, we have one meristematic cell. Yes, we have a meristematic cell. This meristematic cell will divide into two. When this meristematic cell divides into two cells, one cell will remain meristematic. One cell will remain meristematic, but the other cell will become permanent. Will become permanent. Similarly, when this cell divides, this cell eventually divides, one cell will be again meristematic. One cell will remain meristematic, but the other cell will become what? Permanent. It will become permanent. Permanent. Similarly again, if this cell divides, one cell will become meristematic, but the other cell will become what? The other cell will become permanent. This is called as what? This is called as your arithmetic growth. It's called as arithmetic growth. Okay, and this arithmetic growth is very constant. It is a very constant growth. And this type of growth can be found in your roots. The other growth is called as geometric growth. It's called as what? Geometric growth. So in the case of your geometric growth, if this is a cell, this is your meristematic cell. This meristematic cell will divide into, again, both of them will be meristematic here. Both will be meristematic. This will divide into 2. Both will be meristematic again. Meristematic. Divide into 2. This will again be meristematic. Again, this will also will be meristematic in nature. It will be meristematic in nature. Okay. It will be meristematic in nature. That is, both cells, both daughter cells are meristematic. Both daughter cells are meristematic here. Meristematic. And do we have a graph for this? Yes, we have a graph for this. If you look at the graph of your arithmetic growth, can you see here very constant growth? Do you see a very constant growth here? Yes, there is a very constant growth here. We can see a constant growth. Constant growth can be seen here. Very constant growth can be seen here. But if you look at the growth of your geometric growth, there is a small change here. Okay. For example, if you look at the growth of your geometric growth, can you see a sigmoid growth? Yes, it is a sigmoid growth. And this type of growth, the geometric growth, is mainly found in your higher plants. Okay, it's here. Many higher plants and many organs. Now, if you look at the sigmoid curve, we have a first phase called as your lag phase here. Lag phase. Then we have the log phase. Log or exponential phase. Then last we have the stationary phase. When we come to stationary phase, what happens? There is less nutrients. There is very less nutrients, and the cells do not divide further. They die eventually. Okay, this is called as your geometric growth. Easy. Now, are there any formulas here? Yes. Are these formulas important? Yes, they are very important. For example, for your arithmetic growth, we have LT equals L0 plus RT, where LT can be called as your length at time t, also called as your final length. Now, what about your L0, length at time 0? Can I call it initial length? Yes, initial length. Now, what about your R? That is growth rate or elongation per unit time. Now, similarly, we have something called as, for geometric growth, WT equals W0 e to the power of RT. Final size, initial size is going to be W0. R is the growth rate, time, and natural logarithm. This R can be called as relative growth rate also. Okay, R can also be called as your relative growth rate. It's very easy, students, very easy. Not many questions have come from here, but some questions can be seen here. Okay. Now, for example, if you look at your NCERT diagram, do you see under arithmetic growth, one side is always constant? Do you see? One side is always permanent and rest is meristematic. That is, they have capacity to divide. But when we come to your geometric growth, right? Geometric growth, Everything is capable of dividing. All these are dark is dividing cells. Light have lost the capacity to divide. They have lost the capacity to divide. Light, see, cells that have lost the capacity to divide. Dark color one is cells capable of division. All in geometric, all the cells are capable of dividing. All cells are capable of dividing. Okay. Now, when it comes to embryo, it is very, zygote division is very peculiar. Zygote division is a little bit different here. How is it different? When it comes to zygote division, students, the initial stage, do you see this? Initial, zygote to embryo. Zygote to embryo stage. Zygote to embryo stage is always geometric. Geometric growth. Can you see? Geometric growth is there. Then once it forms embryo, later on, it's always arithmetic. It's always arithmetic. Arithmetic. Simple. Simple. Okay. Zygote to embryo, geometric. Embryo later on, it's arithmetic growth. This is your growth. Okay. Now, how do you measure growth? How do you measure growth, students? How do you think we can measure growth, students? How can we measure growth? Anyone in the chat who can tell me? Is there any way to measure growth? Is there any formula? Or is there any way you can measure the growths? One second, students. Is the volume coming right? Is the volume proper, right? Yes. Now, students, I want, if you are able to solve till now, I want everyone to quickly smash the like button first of all. If you're able to solve the, if you're able to understand something, quickly smash the like button. And also, if you want these notes, if you want all of these notes, where will be available? All of these notes will be available on Sir Biology Telegram channel. Will be available on my Telegram channel and also on Vedantu NEET English Telegram channel. All of these notes will be available there. Everything will be available there for free. Okay. Now, how do we measure growth? Growth can be broadly classified, or when you talk to measure it, you can measure it in two different ways. You can measure it in two different ways. One is called as your absolute, other one is called as relative. We have absolute as well as relative. Absolute, we have absolute as well as your relative. We have absolute as well as relative growth. As well as relative growth. Now, what is the difference? What is the difference? Absolute is always, I can say, actual growth. Actual growth. For example, last year I was 60 kgs. Right now I am 65 kgs. What do you do now? All you need to do is final minus initial. That is 65 minus 60. You will tell I have grown by 5 kgs. Right? You do final minus and you get 5 kgs. That is called as your absolute growth, actual growth. But what about relative growth? Relative growth is very simple again. Relative growth is very simple again. That is, it is always in, first of all, in percentage. It's always in percentage. In relative growth, you need to do, let me tell you, quick example. Quick example. Let me give you a quick example, students. Quick example. Is the voice breaking now? Students, is the voice breaking still? Is the voice breaking? No, right? Divya? Naina, Divya, Suhasini, Jai. Is the voice breaking still, students? No, right? Okay. Now, 5 kgs difference is there. Now, if I tell you about relative growth, for example, there is a student A. Student A's pocket money is 5 rupees. From 5 rupees, the pocket money became. Okay. From 5 rupees, the pocket money became 10 rupees. Okay, student A. For student B, the pocket money was 50 rupees. Okay. From 50 rupees, the pocket money became 55 rupees. 55 rupees. But if you do the relative growth, if you do absolute growth, in both cases, can I tell? In both the cases, there is increase in 5 rupees, that's all. 5 to 10, 5 rupees difference. 50 to 55, only 5 rupees difference. That is if you do absolute growth. But in relative growth, what do? In relative growth, we have a formula. In relative growth, we do something called as, let me write here. In relative growth, what do? We do final minus initial divided by initial into 100 into 100. For example, what is final here? 10. 10 minus 5 by 5. That is around 5 by 5 into 100 into 100. That is around 100% increase. 100% increase is there. 100% increase for student A. Now what about student B? That is 55 minus 50 divided by 50. That is around 5 by 50. That is around 10% increase. Just 10% increase. That is called as your relative growth. Growth in a very, when you do relative growth, you know the actual difference in percentage wise, how much of growth is happening. In absolute, it might seem like everything is same in absolute. But when you do the relative growth, you get to know the reality. Here. Okay, get to know the reality here. That is your relative growth. Okay. For example, if you look at your NCERT, this is the diagram given there. For this is one leaf A and leaf B. Leaf A, from 5 it is becoming 10. 5 it is becoming 10. Here it's becoming from 50 to 55. The same concept applies. Leaf A, we have the leaf A and leaf B. Okay. In leaf A, what is the absolute growth? Absolute growth is going to be what? 10 minus 5. Okay. Leaf B, it will be 55 minus 50. In both cases, can I see it's just 5 centimeter square increase? 5 centimeter square is increasing. That's all in both the cases in absolute growth. Now, what about relative growth? Final value minus initial value. That is 100% increase. Here we have what? 55 minus 50 divided by 50. That is just 10% growth. That is your relative for leaf B. So when we do this is the example which is given in your NCERT. Okay. So usually the relative growth will tell you the reality. Relative growth will tell the reality of growth. Okay. Now, there is something called as conditions of growth. Obviously, there might be different conditions which are required for a growth. The first one is obviously your temperature, nutrients, light, water, oxygen. Plants require all of this in a much more precise manner. If something is more or less, plant will undergo in stress. They'll go under stress. So they need all of these properties, water, sunlight, everything in a very fixed amount. Similarly, your water is important for turgidity of the cell. And the same water is also important for all the enzymatic reactions, because all the enzyme, if they want to react, they need a medium, and water is the medium when it comes to cells. And oxygen releasing metabolic energy is there. Nutrition, temperature, light and gravity. Every single thing is required in a proper order. Before we go to the differentiation, redifferentiation and dedifferentiation, let's see the NCERT if you missed out any point. Let's look at NCERT now, all of you quickly. See, this is your growth. This is your growth rate, right? This is your different types of growth rate. Here, growth rate is here. We learned about your arithmetic growth and geometric growth. Arithmetic as well as geometric. In arithmetic growth, one particular is meristematic, other one becomes permanent. Similarly, one cell remains meristematic, other cell becomes permanent. But in the case of geometric growth, every single cell is going to be what? Every single cell is going to be meristematic in nature. Meristematic in nature. That is your growth. Okay. See here. This is the growth when it comes to your arithmetic growth. Arithmetic growth. The example is given somewhere here. Here. Root elongation. Example is given right here. Do you see this? Only one daughter cell contains ability to divide. Exemplified by the root elongation at constant rate here, right here. Root elongation. That is the example for your arithmetic growth. Arithmetic growth. When we look at your geometric growth, this is this here graph. Lag phase, log phase, and stationary phase, as well as your stationary phase. Okay. Leaf we learned, this is your absolute and relative growth. Absolute growth is your measurement and comparison of total growth per unit time is called as your absolute growth, and growth given by system per unit time in common basis is going to be your relative growth. Done. What are the conditions of growth, students? Conditions of growth: plants require water, oxygen, nutrients and elements for growth. By cell size, right? Turgidity of the cell helps in extensive growth. Water is provided for medium for enzymatic activities. Oxygen helps in releasing metabolic energy. We know in respiration we need oxygen. Plants require oxygen. Yes, oxygenic growth. Plants require nutrients, plants require water, plants require right temperature. Plants require, yes, here, optimum temperature they require, and proper gravity. Light and gravity also plays a very crucial role. Light and gravity also plays a very crucial role. And every single point has been covered. Every single point. And trust me, students, after today's class, if you go back and you read every line of NCERT, every line of NCERT will be clear to you, and you will be able to solve every single question. And this is the easy part. Easy part. Just do this for the rest of the year. Every single chapter, learn the concept. Every single chapter, learn the concept, read the NCERT, solve PYQs, do the mock test. No one is stopping you then. No one is stopping you. For example, this de-differentiation, redifferentiation and differentiation. This is a very, very easy concept. They might have given you such a big paragraph here. Let me summarize it for you. Let me summarize it for you in a much more easy manner. Much more easy manner. For example, what is going to be your differentiation? What is going to be your differentiation? Differentiation. In differentiation, we have a cell. We have a mesophyll cell. Let's take it as a meristematic cell, dividing cell, right? Dividing cell. We have a meristematic cell. Meristematic cell. This is a dividing cell. Now, this meristematic cell loses its ability to divide. It loses its ability to divide. It is losing its ability to divide completely. Losing its ability to divide. It will become something called as your differentiated cell. Differentiated cell. And later on it becomes a primary permanent cell. Primary permanent cell. That's all. This is called as your differentiation. It happens normally in all the plants. Okay. For example, is going to be what? Example is going to be primary medullary race. Primary medullary race. Then we also have primary xylem. Then we have primary phloem. Primary phloem. Primary phloem. This is called as your differentiation. Differentiation. Okay. We have a primary permanent cell. We have a primary permanent cell. Now students, now. What happens in your de-differentiation? This is very important. Listen to me very carefully. Lots and lots of PYQs have come from your de-differentiation. What happens in your de-differentiation? In de-differentiation, the primary primary permanent cell. The primary permanent cell again regains the ability to divide. It will regain the ability to divide. It will regain the ability to divide again. It's like, no, I want to divide again. It is regaining its ability to divide, and it makes the secondary meristem. It makes up the secondary meristem. We have secondary meristem now. We have the secondary meristem. Example is going to be what? Vascular cambium. Vascular cambium. We had medullary rays, right? We had medullary rays. The medullary rays give rise to the vascular cambium. Vascular cambium. Now, what about your redifferentiation? Redifferentiation. When we come to redifferentiation, this secondary meristem again will completely lose its, it will lose its ability to divide. Again it will lose ability to divide and form a permanent cell. It will become again a permanent cell. Again it will become a permanent cell. Now this permanent cell will have a specific function. It will have a specific function, very specific function it will have. Very specific function, losing the ability to divide. That is called as your redifferentiation. That is called as your redifferentiation, and it will give rise to a secondary tissue. Secondary tissue. Example is very simple again. Example is going to be your secondary xylem and secondary phloem. Secondary xylem and secondary phloem. Secondary xylem and secondary phloem cannot divide. They have lost its ability to divide, and they have a very specific function. Very specific function. Okay. So what did we start? We started with your differentiation, where we have a meristematic cell losing its ability to divide. It becomes a differentiated cell. That is your primary permanent cell. This primary permanent cell regains the ability to divide. It's a permanent cell, like your parenchyma, collenchyma, sclerenchyma. All of those are your permanent cells. Permanent cell. If parenchyma, collenchyma, sclerenchyma, if it again starts dividing, then it becomes dedifferentiation. It becomes dedifferentiation. Now, it gives us a secondary meristem. Now, this secondary meristem again loses ability to divide and becomes specific function, and it makes the secondary tissue. That is called as your redifferentiation. That is called as your redifferentiation. Please know the difference between differentiation, dedifferentiation and redifferentiation, because you will get a lot of questions. Example, NEET 2024 question. NEET 2024 question right here. It says that formation of interfascicular cambium. Formation of interfascicular cambium from fully developed parenchyma cell. From a fully developed parenchyma cell is an example of. Students, parenchyma is a type of primary permanent cell. If primary permanent cell, if it's again regain the ability to divide, what is it called as? It is called as your dedifferentiation. See, your primary cell is again gaining its ability to divide, and we are getting your interfascicular cambium, right? Here, we are getting vascular cambium also here as a result of dedifferentiation. See, answer is very simple. Very simple answer. Look at this question again. Answer is given here, but don't see the answer now. See, in tissue culture experiments, leaf mesophyll cell. Mesophyll cell is a primary permanent cell. Primary permanent cell put into a culture media to form callus. This is called as again dedifferentiation, because your primary permanent cell is again regaining the ability to divide, and it is dividing. That is dedifferentiation. Dedifferentiation. Look at this question now. The living differentiated cell. We already have a differentiated cell. that lost the capacity to divide anymore. Okay, we have a differentiated cell which has lost the ability to divide. Okay, can regain the capacity to division under certain conditions. This phenomena is called as, for example, we have a differentiated cell. It has lost all of its ability to divide. Again, it is regaining the ability to divide. That is called as de-differentiation. Right? It is called as de-differentiation. See here. Regain the ability to divide is called as de-differentiation. De-differentiation. So students, you will get a lot of questions. I have taken few questions here and there, but you will end up getting a lot of questions from this concept here. A lot of questions from this concept right here. Okay, so learn the concept of de-differentiation, redifferentiation, and trust me, I want you to write and practice all this. Only when you write and practice all this, then only it will stay in your head. Then only it will stay in your head. Otherwise, forget it. Okay. Otherwise, it will never be. You will never be able to remember this. Okay. So always learn, practice. You need to write and practice this. Write and practice this. Okay. Now, can we start development now? Can we start development now? Development, very easy, very easy. Development also is. Students, do you realize your half of the chapter is over right now? Half of the chapter is over right now. See here. We learned about, see, formation, examples are given here. Examples are given here. Differentiation, see, differentiation. Shoot apical meristem. Cells derived from shoot apical meristem and root apical meristem. Root and shoot apical meristem are technically what? Meristematic tissues, right? They are meristematic tissue, right? Differentiation. That is, cambium differentiate and mature to form specific maturation. During differentiation, cells undergo few major structural changes in both cell wall and protoplasm. Okay, to form tertiary elements. Now, de-differentiation is what? Cells that now have lost the capacity to divide can regain the capacity to division under the certain conditions. That is called as a de-differentiation. Formation of meristem, intrafacial cambium and cork cambium. Question will come. Cork cambium and intrafacial cambium are a result of which type of division? De-differentiation, redifferentiation. Okay. So answer should be de-differentiation. Then they can. Okay. Such meristematic tissues are able to divide and produce cells that lose. Then again they lose the capacity to divide, but mature to form specific function. That is redifferentiation again. Then we have redifferentiation. So they keep on changing. Okay. So tell me in the chat. I want you to comment in the chat. Are there any other examples of differentiation, redifferentiation? Let me know. Okay, if you know. Clear? So every single one we are learning, every single point I believe is being covered. We are left with development. Development is a very easy concept. Plasticity, and we'll come to the plant growth regulators. That is the last part of the chapter. Last part of the chapter, which I will give you points. You don't need to read the entire thing. You don't need to read the entire paragraph. I have made some points. Just learn those points and everything will be like a cakewalk. And we're done with the chapter already. Do you realize? Let's understand development. What is development? What is going to be development? Okay. Let's understand development now. What is this development? Write down this definition. All the changes that occur in plants life cycle, is called as development. All the changes occur in the plant's life cycle is called as development. For example, this is the seed. This is a normal seed. The seed will germinate now. Yeah, the seed will germinate into a juvenile. It will develop into a juvenile. The same seed will again develop into a much more developed state. Yes. Can I call it a mature state? In the mature state, there will be development of reproductive organs. There will be development of reproductive organs. Yes. Now, this particular mature state will give rise to your flowering state. Yes, flowering stage. And eventually, once that stage is also over, plant will die. Eventually, the plant will die. This is called as your development. That is, all changes that occur in plant life cycle. If you look at NCERT, this diagram is given. If you look at NCERT, this diagram is given. For example, we have your meristematic stage. A meristematic cell is given here. See, meristematic cell. Cell division, plasmodic growth. Then you have differentiation. Then you have elongation. The cell is maturing. Mature cell, which can produce flowers. Then finally we have senescence, and then we have a death of the plant. This is called as your sequence of development of process in plant cell. This is happening in a plant cell. Now, all of you know the life cycle now. All of you know the life cycle of a particular plant. Now, what if there is any deviation? What if there is a deviation in plant's life cycle? If there is a deviation in plant's life cycle because of certain environmental factors or because of the life cycle of a plant, that is called as plasticity. Any deviation from a plant's life cycle. See, this is the life cycle plant is undergoing. If there is any deviation in plant's life cycle, that is called as plasticity. Okay. So, ability to plants to alter their development in response to environmental condition and also different phase of life. That is called as plasticity. For example, there are two main types here. First one is based on the phase of plant's life cycle. Other one is based on environment. Okay. Change in life cycle of a plant based on the phase and based on environment. For example, based on phase, a plant can different type of leaf in juvenile stage, different type of leaf in adult stage. In juvenile stage, a plant leaf can look like this. In adult stage, the same leaf can look like this. There is a different shape of leaf. That different shape of leaf based on the phase is also as heterophyly. Different leaf forms. Okay. Different leaf forms based on the phase of the life cycle. Juvenile phase, leaf look like this. In adult stage, we have a different phase. Okay. Now, similarly students, based on environmental conditions. For example, if a leaf is in aquatic, it's underwater, the leaf will look like this something. Leaf will look like this. Why? Because there is very less water resistance. Water can flow through very easily. Yes. The same plant in terrestrial ecosystem. If the plant is in terrestrial ecosystem, the leaf will look something like this. Very broad leaf. Why? For photosynthesis region. If our leaf is broad, more photosynthesis, more surface area. So, do you see that based on the environment, life cycle is changing? Change in the leaf shape based on the phase of the life cycle. In juvenile stage, we have a leaf like this. In adult stage, we have a different leaf. Now, how will they ask you the question? They'll ask you the question based on the example. For your change in the life cycle, change in the phase, we have three examples. Cotton, coriander and larkspur. Cotton, coriander and larkspur are the examples for change in phase. But what about change in environment? The change in the leaf shape based on the environment example is buttercup. It is going to be buttercup. Buttercup. That's all. That's all. See here. Change in the leaf based on the stage of life cycle. Juvenile stage, adult stage. Change in the leaf based on the habit, environment, water habitat, so that most of the water can pass through easily. But in it is much more elaborate, more broad, because more photosynthesis has to take place. More photosynthesis has to take place. It's very broad here. So change in life cycle of a plant. This is buttercup. Example for your heterophyll here is larkspur, cotton and coriander. See the question here. Question here. See, this is a NEET PYQ. This is a NEET PYQ. NEET PYQ right here. What does it say? Which of the following plants does not show plasticity? We know cotton shows, coriander shows, buttercup shows. But the maize shows no. Maize does not show plasticity, so option number 1. So the questions are very simple if you know the concept. Just know the concept. You know the concept of plasticity. Plasticity is nothing but change in the developmental pattern. Anything abnormal than the continuous pattern. The life cycle is there. Anything change in the life cycle based on the environment or the phase, that is called as plasticity. Now see this question. It's already given here. See, very easy question again. Ability of plants to follow different pathway in response to environment, leading to the formation of different kind of structures, is called as plasticity. NEET 2022 question. Simple. Right? Based on the life, is there a change in life cycle based on the environment? Plasticity. Okay, plasticity. Now, if you want to divide the entire development, it can be broadly divided into 2 parts. That is growth plus differentiation. Growth plus differentiation. Okay, growth plus differentiation. Now, what are the main factors for it? There are 2 main factors. First one is called as intrinsic factors and extrinsic factors. Intrinsic as well as extrinsic factors. In intrinsic factors will tell, in extrinsic factors it could be temperature, the same things, the oxygen availability, then we have light, gravity, water, the same extrinsic factors. But when we come to intrinsic factors, within the cell, there are 2 main things. One can be called as your intracellular or extracellular. No. Intra and inter. And intercellular. Intercellular. In intracellular, students, in intracellular we have mostly the genetic factors. That is, what are the genome present here based on the plant is able to produce red flowers, green flowers, plant is able to grow this much. That are factors. And intercellular factors are basically your plant growth regulators. They are the plant growth regulators. Those are the intercellular factors. Plant growth regulators. Also called as your plant hormones. What are plant hormones? Plant hormones are basically your chemicals. They are basically the chemicals which regulate the physiological properties. Physiological factors. Just like in case of humans, we have hormones in our body. Pineal gland has hormones, pituitary gland has hormones. We have hormones in our body. Similarly, plant also has hormones. And these hormones regulate the physiological properties such as your cell division, cell elongation, as well as your cell maturation. This is called as your plant growth regulators. And if you look at the plant growth regulators, there are mainly divided into 3 categories. We have something as your promoters, inhibitors. Some of them can have dual function. We can say both promoter or inhibitor, both are there. Under promoters we auxins, gibberellins, as well as cytokinins. Under inhibitors, abscisic acid, ABA, and under both we have the gaseous hormone that's called as your ethylene. We have the ethylene. Clear? Those are the different types of your plant growth regulators. Can we start with auxin now? Can we start with auxin? Students, this is the most important part of your chapter now. Most important part of your chapter. Okay. From here onwards, next 45 minutes are going to be the most crucial ones, the most crucial time. Next 45 minutes, if you concentrate and if you understand the points and remember the points, when I say usually you get 4 questions, right? Usually you get 4 questions every year. Out of these 4 questions, 3 questions will be from next 45 minutes usually. Next 45 minutes, 3 questions. If there are 5 questions, sometimes all 5 of them, all 5 of them will be from this concept alone. So before we start this, I want everyone to refresh their mind. Let's take a small break of 20 minutes. And we'll come back and then we will solve it. Okay? 20 minutes break. Can we start exactly at 2:30? We will start again. Class will start again at 2:30 exactly. Okay. Let's start at 2:30 because I want everyone to refresh your mind, drink some water, eat something, and we will sit again. And next 45 minutes after that, just 45 minutes, students, I want your concept should be cleared after that. You should not tell me, sir, and I have a lot of questions. Students, I have a lot of questions. You see, lot of questions are there. Don't worry. Lot of questions are there. So we will be doing a lot of NEET PYQs. A lot of questions, lot of concepts, lot of information after the break. Okay, so I'll see you exactly at 2:30 and we will start again. Okay. Welcome back, students. Let me know in the chat if I'm audible again, because sometimes what happens is the audio gets disrupted when we take a break. So let me know in the chat quickly if I'm audible to all of you. Yes. Welcome back, everyone. And students, if you have not liked the session, quickly smash that like button right now. And we will start with the last part of the class, which is actually the most difficult. When I say difficult, I mean you have to remember a lot here. And if you open your NCERT right now, there are so many concepts to read. And if you read plant growth part, the regulators from your NCERT, it can get confusing. Let me show you why. Let me show you why, students. It can become a little confusing. See here. So if you read from your NCERT, the plant growth regulators, we're done with this. We are done with the plasticity. Now, do you see this? They have given the discovery, all the discovery in the starting. Entire discovery. See, this is your Fujiko, right? All the discovery in the starting. And then they have given you the auxin separately. Then they have given you gibberellin separately. But trust me, when you are studying, right, you need to study it together. You need to study about your auxin discovery, and then you need to study about the properties of auxin. Similarly for gibberellins, cytokinins, ethylene, as well as your abscisic acid. So that's how I'm going to teach you today the best way to study about your plant growth regulators. So is everyone ready, students? Tell me in the chat quickly if everyone is ready for the last part of the chapter, which is actually the most important part. It is actually the most important part, that is going to be your plant growth regulators. Okay. Now, let me tell you what are the actually important ones here. Every single year, one question will definitely come from your either auxin or gibberellin. One question is always guaranteed from here, always guarantee. Now, let's start with auxin now. Now, who gave the term auxin? Like, where does the auxin word comes from? The word auxin comes from a Greek word. It comes from a Greek word called as auxin. It means to grow. The meaning of auxin is grow. To grow. Auxin means to grow. For example, what is the chemical nature here? Again, students, chemical nature, the discovery, everything is important, and there has been questions here. So remember, the chemical nature is going to be indole compounds. It is going to be indole compounds and precursor. The meaning of precursor is that how are they formed? From what are they formed? Now, what are they formed? So your indole is a amino acid derivative, right? Auxin is a amino acid derivative. It is a tryptophan derivative. And zinc. So auxin is a tryptophan derivative. So I want everyone in the chat to type tryptophan right now quickly. If you're writing the information, write tryptophan. If you're watching it, quickly write tryptophan in the chat, all of you. Quickly, everyone. It is a tryptophan derivative. Chemical nature is indole compounds. This is your indole structure, right? Indole compounds. Done. Now, there are two different types of auxins. One is a natural auxin, other one is a synthetic auxin. The natural auxin includes your indole 3 acetic acid, indole 3 butyric acid, or chloroindole 3 acetic acid. But what is interesting to note here is, this can be written as NAA. Okay, indole 3 acetic acid or NBA. The most interesting part here is this one, 2,4D, 24D and 2,4,5T. These two are your synthetic auxin, and also they are the weedicide. They're what? Weedicide. They help in killing the weeds. Now, I remember a story, okay? During the World War II, all the Japanese were hidden somewhere under the bushes. So what did the US do? US added the weed bomb. They called it as weed bomb. It's called as Project Orange. This Project Orange completely vanished all the weeds from the forest or big plants. If all the bushes are gone, all the soldiers were visible very easily, which are hiding inside the bushes. And they call it Agent Orange. Okay. Now, Discovery is very important again. Discovery is very important. Who discovered your auxin? Auxin was discovered by your Charles Darwin and Francis Darwin. All of you know Charles Darwin, right? Charles Darwin from your evolution chapter. Same Charles Darwin. Okay. Now, plant growth, the topic here is how did the discovery? How was the discovery? The discovery was with the help of a experiment called as phototropism. What is phototropism? Phototropism is nothing but response of a plant to sunlight or light in general. Response of a plant to the light. For example, this is your canary grass. This is your canary grass. Okay, we have the canary grass, students. Canary grass. Now this canary grass, what happens is on this region right here. Do you see? Opposite to the sunlight. Opposite to the sunlight, in this region, there is accumulation of auxin. Accumulation of auxin here. Okay. If there is accumulation of auxin here, this particular region, there will be more growth here. There will be what? There will be more growth here. More growth on this side. More growth on this side. If there is more growth on this region here, if there is more growth in this region, only this side, will it not grow more? This will grow more. So it will start to grow more and your canary grass will bend towards the sunlight. It will bend towards the sunlight. So more growth on this region here. More growth. Similarly, students, they notice that you change the source of light. It will bend towards that. So what did they do is, in one experiment, they completely cut this. They cut this part. They completely cut this part. Now, then they cut this part and then they expose the sunlight. And when they expose the plant to the sunlight, there was no curvature. There was no curvature at all. Curvature was completely absent. It's called as decapitation. Yes, decapitation. Completely gone. Similarly, what they do? One more time, what they do? They took the canary grass and they covered the tip. They covered the tip with a brown paper. Completely covered it. Now, if they cover the plant with a brown tip, will the bending be shown? No. Again, the curvature was, there was no bending. So they identified, Charles Darwin and Francis Darwin, they identified that there is something at the growing tip of the curvature, growing tip of the canary grass, which enables the plant to bend towards the light. That was the discovery by Charles Darwin. But who did the isolation? Isolation of your auxin was done by your FW Wendt. Now how did they do? They took a plant. Awareness sativa. They took a oats plant and cut this part. Okay, they cut this part. Then they took a agar-agar medium. And they placed the cut position into the medium. Okay, they placed the cut position into the medium. This is your agar-agar media. Okay. Now, whatever hormone was present inside the growing tip, everything diffused into the agar medium, into the agar block. It completely diffused into the agar block. Now, this is your canary grass. Again, canary grass. Okay. Now, to this canary grass, they added the block on this side. In one canary grass, they added the block on this side. They added the same block there. They added the block. Obviously, in this region, we have more hormones now. More hormones. In this region, we have more hormones. Now, what happens here? This grass will bend the other way because more growth on this side. Similarly, this grass will bend on other side because more growth on this side. More growth here and more growth on the other side. So this shows us that there is something in the growing tips. Which is able to make sure the plant is growing. Okay, make sure the plant is able to grow. Clear? This is called as A curvature test. This is called as what? This is called as Avena curvature test. Any doubt here? Ask me quickly, all of you. Any doubt here? This is called as Avena curvature test. Done. Now, what are the functions? This is the most important one. Most important one. The first one is root initiation as well as apical dominance. Root initiation is development of roots. Apical dominance is that the plant will grow very tall. There will be not much branching. Branching is not there. It is growing very tall. It is growing very tall. Okay. Feminizing effect, more female flowers. Cell division, cell enlargement, because it is able to make sure the plant is growing. So obviously there will be more cell division and more cell enlargement. Flower initiation. Flower initiation in the case of pineapple. Parthenocarpy, and also prevention of abscission. It is making sure the abscission is stopped. But what is the twist here? There is a small twist here, students. There is a small twist. The small twist is that in your young plants, yes, it is preventing abscission. It is making sure the plant is not dying. It will make sure. But in the case of old plants, in the case of old leaves, especially in the case of old leaves, in the case of old leaf, it will make sure the old leaf will fall. So prevention of abscission will not be there in the old leaf. Abscission will not be there. It will make sure the leaf falls down. Okay. But in the case of young plant, it will make sure the prevention is there. That is auxin. That's going to be your auxin. See this question now. Look at this question from your NEET 2023 question. The phenomena which is influenced by the auxin and also plays a major role in its discovery. Which one is it? Apical dominance, gravitropism, root initiation, or phototropism? Discovery, not isolation. Discovery. Discovery is going to be your phototropism. We learned it. So they will ask you very easy questions. Very easy questions and very direct questions. It's up to you how do you take those questions and if you revise everything. And what is the best way to revise this? The best way to revise this is make a tabular column and write down all the properties. Write down all the properties. I'll show you simple. I'll show you how. I will show you how do you do it. See this. I'll show you how do you do it. See this. This way. You make a summary. Do you see this? Auxin, functions. Gibberellins, functions. Cytokinins, function. Ethylene, the functions. Do you realize? This is abscisic acid, the functions. So when you make such a table, such a tabular format, when you make such a table, it becomes very easy for you to study, revise, and also remember at the same time. So students, don't worry, the entire PPT will be, entire notes will be given to you. So you can revise, but I would advise is make your own notes out of these notes. Do a self-study and make your own notes. Okay, make your own notes. Now look at this question now. Now we will do gibberellins. Again, gibberellins is very important. Lot of questions come from gibberellin. What is the chemical nature? The chemical nature is terpenes or terpenoids. What was the chemical nature in the case of indole? So in the case of your auxins, the chemical nature was indole. Now what is the precursor? Precursor is acetyl-CoA and your malvonic acid. Acetyl-CoA. What about in the case of auxin? In the case of auxin, the precursor was tryptophan. It was tryptophan. Now, discovery was by Kurosawa. He was a Japanese scientist. Who, in his field, he observed that. There is a plant which is your mostly wheat plant. It is growing very tall. The plant is growing very tall, but it is not producing any seeds. Is it any of any use then? Imagine a plant is growing very tall and it is not giving any fruits or any seed. It was not useful to the farmers. So he called this plant as, or he called this rice plant as Bakane disease or foolish disease. Now why is it called a foolish disease? Because I told you, the plant is growing very tall. But it is not providing any kind of seeds or fruits. So Bakane foolish disease. And later on, then he observed why is it happening. It was happening because of something called as a fungus, Gibberella fujicori. It was happening because of a fungus called as Gibberella fujicori. It was growing in the stems, and because of this fungus, it was making sure the plant is producing more and more one hormone. That was your gibberellins. Okay, so isolation was done by your Yabuta and Sumuki by is called as alpha amylase Barley endosperm test. Now, you might ask me, sir, is there only one type of gibberellic acid? There are many types of gibberellic acid. In your isolation, there are more than 100 types of gibberellic acid been reported. More than 100 types of gibberellic acids have been reported, and the most common one is your A3. A3 is the most common one. Gibberellic acid A3 is the most common one. Okay, and it is also the first gibberellic acid to be discovered. What are the characteristics? Obviously, it's called as gibberellic acid, so it has to be an acid. Okay, transported either through xylem or phloem, formed in the plastids. It is formed in the plastids. Apart from that, every function is very important here. That is, it stimulates the stem growth. That is, this is the stem. This is the normal stem. When we add gibberellins, the same stem will what? Elongate. The same stem will elongate with the help of gibberellic acid. Increase in stem length. Then we have seed germination. Very important one. We will come back to this again later on. Promotes seed germination. Bolting of rosette plants. Now, what is this bolting? Rosette plants are like your cabbage, cauliflower plants. They're in a rosette form, right? So you can see bolting there. You can see a bolting. You can see the elongation of a stem here. Elongation of stem. Delays senescence. It is like, no. Wait. Time is there for dying. You wait for some more time. So it is delays senescence, bolting in rosette plants, promotes seed germination, and stimulates cell elongation. Apart from that, there are other functions which are more important. That is, increasing the length of grape stalk. For example, this is your grapes plant. Now, if you add gibberellins, what happens? If you add gibberellins, the entire stem gets elongated. So the grapes will be, the entire length will be super high. Do you see that? The entire grapes stalk of the stem becomes more. Okay, increase the length of the grape stalk. Increase the length of the stem. Obviously, we learned that. The best example is in sugarcane. Obviously, students, if the sugarcane length is very high, the stem length is very high, more sugarcane yield. Yeah, because we derive the sugarcane juice from the stem itself. So, example for your length is sugarcane increase the yield as much as 20 tons per hectare. Elongates and improvises shape of the apples. Again, this is a PYQ. Speeding up the malting process in your brewing industry. Spraying, if you add spray of your gibberellins on juvenile conifers. In juvenile conifers, if you add the spray, it becomes mature. It becomes mature very fast. Students, if you read the entire NCERT, you will have a lot of points which you cannot write down properly. And I have written it for you right now. So make use of this, take an advantage of this, download the notes, study every single day, make a summary. Because questions will be easy and direct. Look at this question now. Spraying sugarcane crop with which of the following plant growth regulators increases the length of the stem, thus increasing the yield? This is a NEET 2024 question. This year question. Easy 4 marks, can I say? Can I tell easy plus 4 marks? Yes. What is the answer? Obviously, gibberellins we just study. So, questions will be easy. It's only a matter of if you remember the points, remember the function of every single different types of plant growth regulator. And the only way you'll remember it is by writing many times as well as revising it many times. Make a table, revise every single day. Okay, until you remember everything. Look at this question now. Spraying of which of the following phytohormone on juvenile conifers helps in hastening the maturity period that leads to early seed production? Didn't we just read one line under your gibberellins? Spraying on conifers, conifer becoming maturity is under gibberellins. Gibberellins is the answer. Done. Look at this question now. Answer is given here. Plant hormone which is used to destroy weed in the field. I told you, weedicide. Now tell me, is this 2,4-D? Is this 2,4-B? Is it a auxin or gibberellin? Tell me in the chat. Auxin or gibberellins? Is it a auxin or is it a gibberellins? Quickly. If it is an auxin, is it a natural auxin or is it an artificial auxin or synthetic? Obviously, it is a synthetic one. Yes, it's a synthetic auxin. Easy question. This also is from a NEET 2021. NEET 2021 question. Okay. Now, two hormones are done. Two hormones are done now. Do you realize that? All you need to do is remember the properties, remember the precursor, remember the chemical nature, remember the discovery. And if you read NCERT, it can get confusing. Download the notes. Where do you find the notes, students? I'm repeating again. Notes will be found on my Telegram channel. Biology. Notes will be found here. And also Vedantu NEET English Telegram channel. Download the notes. It's going to be very easy. Okay. Now, cytokinins. Cytokinins, chemical nature is very important because there has been a question. The chemical nature is going to be purine derivative. Very important. It's going to be adenine. Please remember this. The chemical nature of your cytokinins is purines, adenine. Precursor tRNA, discovery 𝐂𝐤 and his co-workers, isolation by Miller. Discovery 𝐂𝐤, isolation Miller. Don't get confused. Do not get confused. Okay. Now, how did 𝐂𝐤 discover? When he was doing experiment. When he was doing experiment on your tissue culture. When he was doing tissue culture experiment, he added onto a test tube. Onto a test tube, he added extracts of your vascular tissues, yeast extract, and coconut milk. These three different types of extracts plus auxin he added. Okay. When he added only auxin, the callus formation was not very proper. Not very proper growth. But along with auxin, when he added your extracts of your vascular tissue, yeast extract, and also coconut milk, there was a proper callus formation. So he noted that 𝐂𝐤 and his co-workers, they noted that there is something in the extracts of your vascular tissue, yeast extract, and coconut milk, which is making the callus grow properly. That is, 𝐂𝐤 was working on tobacco callus. He observed that tobacco callus proliferated on addition of auxin along with auxin when he added any of the three following. And later on, Miller isolated it. Miller, from your herring sperm whale, there is a whale called a herring whale. From the sperm of the whale, he isolated the cytokinins. That is, DNA showed maximum cell division. DNA is powerful. Miller et al. Later identified the crystalline cytokinins promoting the substance that has term called as kinetin. So he observed in the herring sperm whale, he got the kinetin. And he noted that, kinetin is able to do cell division very well. Similarly, in the case of maize, he obtained something called as zeatin. So cytokinin along with auxin promoted the proper cell division. So they noted that there is something which is able to divide cells very well when it is with auxin. And they finally identified the cytokinins. Same with coconut milk also, cytokinins. Now, what are the functions here? Obviously, the main function of your cytokinins is cell division. Modifies apical dominance and promotes lateral growth. Can I tell now? It is antagonist to your auxin somehow. See, auxin is promoting apical dominance. apical dominance, but your cytokinase is promoting lateral growth. Lateral growth. Delays senescence is like weight again. See, promoter. All of them are growth promoters. Promote nutrient mobilization, promotes chloroplast development, helps production of new leaves and adventitious shoot. Adventitious shoots. Look at this question now. Look at this question right here. Which of the following growth regulators is an adenine derivative? See, direct question. And if you have learned from my class, you will be able to answer any of the questions from your plant growth regulators. If you have these notes, you will be able to solve every single question. Every single question. For example, answer is very simple here. That's going to be your cytokinins, adenine derivative, adenine purines, right? Now, that was your growth promoters. Auxin done, gibberellins done, as well as your cytokinins are done. Chemical nature important, as well as your precursor is very important, and functions are very important. 3 points, everything is given in detail. You cannot go anywhere wrong. You cannot go wrong anywhere. Okay. Now, we will start with your ethylene now. Ethylene. Now, first of all, let me tell you, ethylene has a dual function. Dual function. So, in ethylene, you have a lot of functions to remember. You will have a lot of functions to remember. But I have categorized them so you can remember it very well. Don't worry. And next thing you need to remember is ethylene, since it has a dual function, it is also a gaseous hormone. It is also a gaseous hormone. Gas it is, gas. Okay. Ethylene is a gaseous hormone. Precursor is a methionine. Again, do you see that? Methionine is also a type of your amino acid. Methionine. So they can ask you a question. Which are the two hormones which are amino acid derivative? You should be able to tell. So tryptophan and auxin and methionine comes to ethylene. Two of your amino acid derivatives. Amino acid derivatives. Discovery was H.S. Cousins. Bioassay is triple response test. Now, functions. Functions are very discrete, students. Functions are a lot of functions here. So I've divided the functions based on where are they functioning. For example, effect on seed and seedlings, effect on shoot, effect on root, effect on your flower, right? Effect on your fruit, as well as your effect on various organs. So I have divided them based on where are they affecting. Where are they affecting? So it's going to be easy for you to remember it also, right? Now, for example, on seed and seedling, how are they affecting? How are they affecting? It breaks seed dormancy. If it is breaking seed dormancy, it is promoting germination. Done. Horizontal growth of seedlings, swelling of embryonal axis, and it does apical hook formation in dicot seed. Apical hook formation. Okay, the apex, growth of apex. All of these are effects on the just seed and seedling. What about on the shoot? Obviously, it breaks bud dormancy, sprouting of potato tubers, internodal and petiole, very important, see, very important. Internodal and petiole elongation. Petiole elongation in deep water rice plants. This is for shoot, only for shoot. What about in the case of root? That is, promotes root growth and also promotes root hair formation, very important. Because when we think about root growth, always and always auxin comes to our mind. Auxin promotes root growth. But what about root hair? When you think about root hair, it's always going to be your ethylene. Ethylene promotes root hair development. Ethylene does root hair development. Then you see flower. Induces flowering in your pineapple and mango. Promotes, see again, very important line. Very important line. Promotes female flower in cucumber and hence increases the yield. More flowers in cucumber or cucumber. What about in the case of fruits? Commercially used for ripening the fruits. It is used to ripen many fruits. Ripen fruits. And how? I didn't tell you to discover, right? And ethophone. Ethophone, source of your ethylene. Plant convert the ethophone to ethylene. Okay, so they help in ripening of the fruit. So if they now ask you the functions of ethylene, don't tell me the fruit. There should be effect of ethylene on shoot, root, seed, as well as your fruit. As well as your fruit. And students, remember, it was the same fruit. It was the same fruit which helped, to find the bioassay. something called as triple response test. Let me tell you how it was. Bioassay. Bioassay was very simple. They took an orange, and they kept raw bananas. Raw banana. Some gaseous hormone from the ripened orange was gaseously spent to your raw banana. Now, because of this gas, raw banana became a ripened banana. A ripened banana. That is called a triple response test. Okay. Now, that was your effect of seedling, effect on shoot, effect on root, effect on flower. Flower, remember, two: pineapple, mango, and cucumber. Flower, as well as your fruit, ripening of fruit. Now, usually it does senescence and abscission. That's why it's called as growth inhibitor also. Okay. Now, they'll ask you a question like this. Direct question. See the question. The hormone which promotes internode and petiole elongation in deep water rice. Now, when you read this question for the first time, you might think it is gibberellins because there is elongation of petiole or something. You might think. But if you have my notes and if you have learned from me, you should be tell, sir, deep water rice, ethylene. It's going to be ethylene, right? Ethylene. Many students would go for gibberellic acid. It's actually wrong. It's going to be your deep water rice plant. It's going to be ethylene. Okay. Now look at this question now. Gaseous plant hormone regulator is used in plants for. What is it used for? Kill dicot weed? No. That is your auxin, weedicide. Speed up the malting process? No. That was your gibberellins. Promote root growth and root hair formation to increase the absorption of surface? Yes. Root hair formation. Do you see? Root hair formation. Ethylene. To help overcome apical dominance? No. Okay. That is your cytokinins. Okay. Look at this question now. Production of cucumber has increased manifold in recent years. Application of which of the following phytohormone has resulted in the increased yield of the as hormone is known to produce female flower in the plants? I told you, for the flower, pineapple and apple is there. Pineapple and banana is there. Also cucumber. So it is producing more and more flowers in cucumber, female flowers. Easy question. And this also right here is a NEET PYQ, students. Again, it is a NEET PYQ. Okay, NEET PYQ. Ethylene. So do you realize, are the questions easy? Tell me in the chat now. Honestly, tell me in the chat. Are the questions easy? Tell me honestly in the chat. Are the questions easy and very direct questions? Are they very direct questions now? Easy and direct questions? Yes. The only way you will realize there are easy and direct questions if you solve them. Sit and solve them. And if you know all the functions, know all of these functions, and nothing can stop you from scoring those easy 16 marks. Easy 16 marks. Right? And trust me, students, unless you remember them, all these are NEET PYQ. I'm not giving you easy questions deliberately. All these are very, very easy questions, PYQ questions. Okay. The last one is called as your abscisic acid. Chemical nature is carotenes. Precursor is violozanthin, which is a type of xanthophyll. Discovery was given by your three different scientists. Many different scientists are there. There are three different scientists who are working separately. Some of them called it something called as dormin, which is suppressing the growth. Abscissin 2, which is suppressing the growth, as well as your inhibitor beta. Three scientists discovered this, some type of chemical which is inhibiting the growth of the plant. They call it different names. Later on, after many years, we got to know that all three of them, dormin, abscissin 2, as well as inhibitor beta, everything is what? Everything is abscisic acid. Okay? All of them are abscisic acid. Later on we got to know. Okay? Functions very important again. Increases the tolerance to stress. Stress plant is also called as stress hormone. General plant growth inhibitor, it is inhibiting the growth. Inhibitor of plant metabolism, seed development and maturation. Now, it promotes seed dormancy and inhibition of germination. It is inhibiting the germination, inhibiting the plant germination. It is inhibiting. But did we learn some hormone which is promoting germination? Think again. Under gibberellins, did we learn that? Under gibberellins, did we learn that? Where is gibberellins? Gibberellins here. See, promotes seed germination. Gibberellins promote seed germination, but your abscisic acid is inhibiting it. It is completely inhibiting it. It is inhibition of germination, inhibiting germination. See here, stimulates the closure of stomata. Yes. Can I tell it is antagonist to gibberellin? It is against gibberellin, working against gibberellin. Yes. So in your examination, if you get a question, which hormone is antagonist to gibberellins, your answer should be abscisic acid. Abscisic acid. Okay, abscisic acid should be the answer. Now look at the functions. Everything is given here, students. Every single function is given here, and you can go through this. It's going to be very easy for you to understand. See, cytokinins, listens, promote nutrition mobilization. Everything is given here. Everything is given here. Okay. So if you know that, let's solve this question now. Let's solve this question. Auxin is used by gardeners to prepare weed-free lawn, but no damage is caused to the grass of the auxin. It is no damage is caused to the grasses, as auxin does not harm your monocots. It does not harm your monocots. Here, it's a line of NCERT. I'll show you. It is a line of NCERT. I'll show you. Okay. Now match the following. You do match the following. So all of you know. What about gibberellins? Tell me. What does gibberellins do? Gibberellins will do what? Increase the stem elongation. So D is 1. See, just where is C? D is 1 here. 2 is the answer. That 2 is the answer. Ethylene does what? Ethylene does promotion of your senescence and abscission, dual function, right? And your cytokinins will do what? Cytokinins will delay leaf senescence. And then we have ABA, inhibition of seed germination, antagonist germination. Done. Easy question. Look at this question now. Which hormone is used to induce immediate stomatal closure in leaves? Didn't we just learn? Stress hormone. Which is that? The stress hormone is nothing but your abscisic acid, which will close the stomata whenever there is stress. Done. This is your homework question, students. This is your homework question. This is your homework question. What does it say? The phenomena by which undividing parenchyma cells. Parenchyma cells are what? Primary permanent cell. Primary permanent cell starts to divide mitotically. That is, they regain the ability. They are regaining the ability. During the plant, tissue culture is called as. What is it called as? Very simple. Very simple. That is a homework question. Let's just go through NCERT if you don't miss any. If you did not miss anything. Let's go through it once. So, growth regulators. See, plant growth regulators are small, simple molecules and chemical composition. They can be indole compounds, indole 3 acetic acid or IAA, right? IAA. That is your auxin. They can be, see, they have given you everything here. They have given you everything here, but they are not telling you from where is getting it from, right? Are they mentioning auxin anywhere? No, they are not mentioning auxin anywhere. Didn't we learn adenine derivative? Adenine derivatives we learned. N6 purines, kinetin, abscisic acid, ABA, terpenes. Then we have grasses, ethylene. Plant regulators are variously described as plant growth substances, plant hormones or phytohormones in literature. Okay, G3, that is gibberellic acid, the first one to be isolated. So the chemical nature is given right here. The chemical nature as well as sometimes even precursor is given here. Precursor. Okay, and you have with the help of notes you have written now. Okay, now PG are broadly classified into two groups. That is, one group of PG are involved in growth promoting, that is your auxins, gibberellins and cytokinins, flowering, fruiting and seed formation. They are also called as plant growth promoters, auxin, gibberellins and cytokinins. The PGR of those groups play an important role in plant response to the wounds and stress of biotic and abiotic origin. They are involved in the various growth inhibitors. So stress is inhibitor, dormancy and absorption. The PGR abscisic acid belongs to this group. The gaseous PGR ethylene gaseous could fit either in groups, that is, it is both dual function, but it is large in inhibitor activity. Now they have given you, see here, they have given you the bio discovery. Interesting discovery of each of the 5 is given here. See, Darwin and his son Franz Darwin, they did what? Avena curvature trough, Avena curvature, photoperiodism. We learned this photoperiodism. They cut this curvature. Isolation by FW went, that is tips of coleoptile of oat seed. See, isolation by FW went in the oat seed. Okay, in canary grass by your Charles Darwin and Francis Darwin. Charles Darwin as well as Francis Darwin. Okay, now Bakane disease by your Fujikori onion, by a disease caused by your fungus that is Gibberella fujikori by Kurosawa. Okay, in the rice plant, the plant kept on growing but no seeds. Skoog and his co-workers observed that the internodal segments of tobacco stem, that is callus, a mass of undifferentiated cells, proliferated only if, in addition to auxin, the nutrient medium was supplemented with one of the following extract. That is, along with auxin, if they added something else, that something else had cytokinins. They had cytokinins. Extracts of vascular tissue, yeast and coconut milk or DNA. Miller later identified, that is isolation, and crystallized the cytokinins promoting the active substance they termed kinetin. Then we got in 1960, 3 independent researchers reported the purification of chemical characterization of 3 different kinds of inhibitors. 3 different inhibitors. But later we got to know everything is same. Everything is same to same. Everything is same to same. See here, inhibitor B, abscission 2 and dormin. Later all 3 were provided to be chemically identical, named abscisic acid. H.S. Cousins confirmed the release of volatile substance from the ripened orange. Did we see that orange is ripening a banana here, unripe banana into volatile substance was identified as ethylene, the gaseous PGR. Auxin, we learnt all the functions given here. See, all the functions are given here. In auxin, from the Greek word auxin, first isolated is indole 3-acetic acid. That is your natural one. Synthetic also, synthetic is what? Synthetic is your IAA, IBA, 2,4-D, right? 2,4-D. All of these auxins have been used extensively in your agriculture and horticulture practices. That is weedicide. They help in initiation of root. Initiation of root is auxin. Initiation of root hair is ethylene. Auxins promote flowering in pineapple. We wrote that. They help in preventing fruit and leaf dropping at early stages, but promote action of older mature leaf and fruit. So yeah, leaf and fruit it will protect. Auxin will protect. But if the leaf is very old, they like go die now. In most higher plants, the growing apical bud inhibits the growth of the lateral buds. Phenomenon is called as apical dominance. So apical dominance by auxins. They'll make sure the lateral is not growing at all. Okay. Here also, see. It is widely applied in the tea plantation, your hedge making. In hedge making, they do make sure that, so the hedge making they do, you have seen the tea plantation in Assam, everything will be in same level. They make sure that everything is same level so they can pluck the leaf very easily. In hedge making. Okay. Now see this. Auxin introduced parthenocarpy, tomato, widely used in herbicide, 2,4-D herbicide I told you. Kill dicot. See here the line is here. It widely used to kill dicot weeds, but does not affect the mature monocot plants. At all. Also helps in control xylem differentiation and help in cell division. Now gibberellins, GA1, GA2, GA3. GA3 was first discovered, gibberellins to be discovered and remaining most of the intensive study in your all forms. Functions is given, you see. Increase in length of the axis used to increase the length of grape stalk. Fruiting like apple to shape, improve the shape. They also delay senescence. The fruits can be used to speed up the malting process in the brewing industry. Every single point is mentioned. Sugarcane store carbohydrates. So they increase the length of the sugarcane. Done. Spraying juvenile conifers with GA hasten the maturity. Done. Introduce elongation just prior to flowering in beetroot, cabbage, and many plants with rosette. So it does what? Bolting in rosette. Bolting in rosette. Promote bolting. That is intermediate elongation, intermediate elongation. Cytokinins discovered by kinetin, a modified form of adenine. See, they were discovered as kinetin in plants. Kinetin. Okay. Autoclaved hereby. See, autoclaved herring sperm DNA. Kinetin does not naturally occur in plants. Oh, there is zeatin. Where is zeatin? Zeatin is plants. Yeah. Since discovery of zeatin, which is in plants, naturally occurring cytokinins and some synthetic compounds are also done. Rapid elongation function is apical development of shoot, young fruits, new chlorophyll, new leaves, chloroplast in leaves, lateral growth development, everything is given here. Ethylene is also given here. Ethylene, again, different. Influencing ethylene on the plants induce horizontal growth, seedlings swell up of the axis, apical hook formation, everything we have mentioned. Ripening in fruits, leaves done. Break seed dormancy, initiation of germination done. See here, internode and petiole elongation in deep water rice plants done. Root hair and root growth and root hair development, very important line. It is does in flowering, also done. Etaphone, which is used for your development of your fruits. Accelerate abscission in flowering and fruits. Thinning of cotton and cherry and walnut. Promotes fleaway flower in your cucumber, therefore increasing the yield. PYQ again, PYQ from every line. Abscisic acid inhibitor everything. Under your stress conditions, closing of your stomata and increasing the tolerance of plants to various kinds of stress. Stress hormone. Antagonist to gibberellic acid. Antagonist to gibberellic acid. Done. I think we have finished everything. We have finished everything. Every single line of NCERT has been covered. Students, that is for today. I believe the chapter is over. Your entire plant physiology over. Now I would advise you to quickly smash that like button again and share this video with all your friends because it is a very important video and this chapter holds around 16 to 20 marks and I want no one to miss out on this marks. Okay. And also let me know in the comment section how was today's session. Did you understand every line of NCERT? Were you able to solve the questions and were you able to understand the concepts today? Understand the concepts. And students, if you are not a part of DNA batch, I would advise you to join the DNA batch because in DNA batch we're doing beautiful. And anyone who is from the DNA batch, they'll tell me right now, sir, it has been doing really great at every DNA batch. So with that being said, I'll take off right now. I would advise you to read the NCERT on your own, make your own notes, download these notes, right? Already download these notes also. That is also very important. So take care students, bye bye all of you and have a great day. Okay? Have a great day. Let me know in the comment section. Let me know in the comment section with the help of homework question and also how was the session today? How was the session? Okay?